This video is going to have a look at genetic organisms, in particular how bacteria can be used to produce human insulin and how it can be used to genetically modify plants. It's also going to focus on the advantages and disadvantages of using these microorganisms throughout the world. So first things first, then we're going to have a look at how bacteria can be used to produce insulin. Now this is something you've done before, but you need to know it in a little bit more detail. The first step is to remove a plasmid from bacterium. Now that plasmid is a loop of DNA you should be able to recall. As soon as that's been done, the insulin gene is cut out of the human chromosome using something called a restriction enzyme. Now that restriction enzyme recognises the specific sequence of DNA and decides where it needs to cut. So once the restriction enzyme has recognised that specific sequence, that is the human insulin gene, it will cut the DNA at these points, and it will leave at each end an unpaired base, and that is something called the sticky end. The next step then is to do exactly the same to the plasmid. So they'll use the same restriction enzyme and they'll cut it open, leaving the same sticky ends, just obviously the opposite bases. So next they'll take the plasmid and they'll take the insulin gene and they will mix them together. And then they'll need to add an enzyme to it to join them together, and this enzyme is called ligase. This joins the sticky ends together and produces recombinant DNA, so two different bits of DNA stuck together. They'll then take this recombinant DNA and insert it into a bacterium. The modified bacterium then is grown in a fermenter, as you've seen in the biotechnology video, and you can end up with millions of bacteria that all produce insulin for people with diabetes. This second part of the video then is going to have a look at Agrobacterium Tumefacien, and I did have to look up how to pronounce that, and how it's used to genetically modify plants. Now this bacteria, which I'm not going to pronounce again, is a pathogen that invades plant cells and inserts its genes into the plant's DNA. Now the beauty of this is, if other genes are added to that bacterium using the same genetic modification that you looked at a second ago, then those genes are taken along too. This bacterium is known as a vector, and a vector is a carrier that's used to insert DNA into other organisms. So how is it done? Said the examiner. Well, here we go. The key example is for herbicide resistance in a plant. So if you've got a plant that already has a resistance to herbicide and you can work out what gene is responsible, what they can do is they can take that gene from the plant and then they can take this bacterium, the Agrobacterium Tumefacien, and take a circular loop of DNA out of it and then cut it open, inserting your herbicide resistance gene. You then combine it all together, put it back into the bacteria, and then let it infect the cells of a target plant. Once the plant is infected, the gene for herbicide resistance will be inserted into the plant's DNA. This will then grow and grow until the plant itself becomes herbicide resistant. So what are the main reasons that we want to genetically modify our crops then? Well, one of the main reasons is in particular to make plants resistant to insect pests. So scientists discovered there's a bacterium called Bacillus thuringiensis which produces a toxin, and that toxin can kill off many of the insect larvae that are harmful to crops. So what scientists did was take that gene for Bt toxin and inserted it into the different crops like corn, cotton and so on. This then produces the toxin in the stems and leaves, making them resistant to the insect pests. Now the huge benefit for this is that it's harmless to humans, animals and other insects. But because we've only seen the short-term effects of this, we're not 100% certain of the long-term effects of this exposure to these crops. And likewise, because the insects are constantly exposed to this toxin, we don't know how quickly they'll build up a resistance and not be killed by it, so we try to avoid this by using other insecticides as well. So let's have a look at the pros and cons of using these genetically modified organisms. Now one of the main pros that we just looked at is making crops that are resistant to pests. This leads to an increased yield of crops. You can also engineer crops so they can grow better in conditions that they're not used to, so for example in drought conditions. Again, this improves your yield. And last but not least, in the pro column, some crops can be engineered to combat certain deficiency diseases. So for example, if you've got someone that doesn't get enough vitamin A, what they can do is produce something called golden rice. Now that's been genetically engineered to produce a chemical that is converted in the body into vitamin A. There are definitely some cons though to this. and one of the key ones is that many people argue that people get so hungry because they can't actually afford to buy food, not because there isn't any about. So they say that we need to have a look at poverty first and see if we can tackle that. They also say that there are fears that the countries can become dependent on the companies that sell these genetically modified foods. And finally, the soil can be so poor that even genetically modified crops can't survive in it. The final section of this video then is going to have a look at how developed countries can use genetically modified food as well. So snapdragon flowers, which look like this, contain something called a flavonoid. Now a flavonoid is a molecule found in many plants that has an antioxidant effect. These are thought to protect against cancer and heart disease. So scientists then, they took this flavonoid gene and did genetic modification, similar to the way that you've seen it previously, and they put it into tomatoes. A weird side effect of this is it turned the tomatoes purple, as you can see here. The benefit of this is when people eat them, it gives them that antioxidant effect which has been linked to protecting against cancer and heart disease. So apart from the fact that they've turned the tomatoes purple, it can slightly change the flavour of the tomato, which some people obviously won't like. And there are other people that are worried about the long-term effects of these GM crops, both on people and on farms' biodiversity, and whether these genes can get out into other plants.